The third speaker from the University of Sussex, Dr. Eva Hoffmann, Senior Research Fellow and EMBO Young Investigator at the Genome Damage and Stability Center, University of Sussex. Dr. Eva received her PhD in biochemistry from University of Oxford in 2002, and also go on to do postdoctoral research, become a postdoctoral research associate at the University of Leicester in the UK. And currently, Dr. Hoffman's research interest is in protein that promote crossing over, meiosis, and mitosis. Her laboratory recently identified function of the crossover promoting protein MSH4 and MSH5 and the SIG1 in the segregation of the chromosome pairs during the first meiotic division. And the regulation of this process is very important as defective segregation that can lead to birth defects as well as infertility in human. And today we are very lucky to have her here with us today, and her title of the talk is Toward an Understanding of Human Aneuploidy. Please welcome Dr. Eva Hoffmann.
Okay, thank you very much for having us here. We're really having a great time. You've heard from two of our most distinguished scientists at the Genome Center and within the School of Life Sciences, Professor Lehman and Professor Collicutt, giving you an overview, extensive overview of their many, in some cases, decades of research within DNA damage. What I'm going to do as a young investigator now is give you about 10 minutes of what I'm trying to do and set up, what we're doing towards it, and then give you an overview of that the MRC is actually celebrating its centenary this year. So the Medical Research Council in the UK is one of the oldest publicly funding agencies that fund a lot of the research that we do at the School of Life Sciences. I'm directly funded by the MRC, who gives me quite a bit of funding and my own personal salary as well. And it's celebrating its 100 years of discovery. So I'm going to give you a bit of an overview of the MRC as well and the kind of research that the MRC does more generally. So in the lab, we're trying to understand how human aneuploidies arise. And the type of aneuploidies that we focus on are those that are congenital, meaning that although some aneuploidies within the human body are clearly drivers for development of cancer, a lot of the aneuploidies in human that are important, they are congenital, meaning that they occurred from birth. And the most common ones that you see here are the trisomies, Downs, Edwards-Patel, you've probably heard of. Those are the ones that we screen against in doing antenatal pregnancies. Monosomies in human are unviable, except for the sex chromosomes where Turner, for example, they are viable. Klinefelters have problems, but they're quite prevalent too. So the autosomal trisomies in human, the three most common ones, are the major cause of pregnancy loss in human. About of the lost pregnancy that occur, we think about at least 50% are due to extra supernumerary chromosomes. They're also the major cause of congenital birth defects as well, and the major cause of intellectual and developmental disability in human more widely. So where do these aneuploidies and the extra chromosomes originate from? So when there are two possible causes. One is during mitosis, when the zygote starts to develop into a human or into a human baby, and the other possibility, of course, is during meiosis. And the two cell cycles are very different. In mitosis, I'm showing you here two chromosomes. They're diploid, same pair, one from mom and one we've inherited from our dad. Cells undergo DNA replication during S phase, and then during mitosis, the sister chromatids, they come apart, and eventually yielding two diploid cells, daughter cells, that are similar to what the starting material was. So this is kind of a proliferative cycle. In contrast, the cycles that are extremely important for the evolution and of our species is meiosis, and that's the production of gametes. And as you can see here, It starts out very similarly with the development of sister chromatids doing DNA replication, but then homologous chromosomes, they pair, they find each other, and then they segregate away from each other at meiosis 1, yielding these two haploid cells that eventually then go on to divide again, giving rise to these haploid gametes that only have half the genetic content compared to the starting cell. So when we look at the origin of the human trisomies and where they come from, we can trace them back to defective meiotic chromosome segregation. And the way that this has been done is a multitude of ways. First of all, in population studies that started by looking at trisomy families, Down syndrome was in the US, by looking at the genetic information contained on the extra chromosome, it was found that 90% was transmitted from the mother. Fewer than 2% of the Down syndrome patients were mosaics. And that means that it came from meiosis as opposed to doing divisions when the fetus was formed, because then you would expect different tissues to show different amounts of trisomies. We can also look in clinical material from lost pregnancies, and of these embryos and fetuses that have been lost, again, the ones that are aneuploidy, 90% come from the mother. And then finally, if we trace it back to gametes, you can see that when you look directly in oocytes from women who are undergoing IVF, not necessarily because they can't conceive, but for other reasons as well, we find that about 30 to 70% are aneuploid, depending on how you measure this. In contrast, when you look at sperms from clinics, we're seeing fewer than 1% are aneuploid or have the incorrect chromosome number. So from a genome stability point of view, human oocytes are really, really bad at regulating their chromosome content. Okay, so what are the factors that contribute to this problem? And I think the best recognized factor generally is age of the woman. So if you look at the maternal age here, that's been binned, and then you look at the incidence of trisomies, and these are just of clinically recognized pregnancies, you can see here that there's a quite sharp inflection point at around 35 years of age, and then you see an exponential increase in the trisomy incidence as the woman ages when she has the baby. Okay, so this is an increasing problem for health, for our reproductive health, because of the demographic changes that we're going through in reproductive choices. So in the UK in 1992, there were 18,000 cycles of IVF. In 2007, that was increased to 46,000. And I think more worryingly, when you look at the age distribution of women undergoing IVF, you can see in 1992, the median age was around 33, 34. That's now shifted to about 36 plus by 2007. So it's not as easy as just simply looking at age. There are also chromosomal differences. So not all the chromosomes are the same. And that, I think, the past 10 years of research has found that that's actually quite surprising. We used to think that all chromosomes would be the same, but that's certainly not the case. If you look at the maternal age curve down here and the incidence of trisomies, what you find is that, for example, chromosome 16, you see a pretty linear increase with age. Whereas in contrast for chromosome 18 and 21, they show the more classical exponential increase as the woman gets older. So that in itself is actually quite puzzling. When we look then at the other very predisposing factor that we know of, we look at the association of recombination. What we find is that when homologous chromosomes have crossed over, there tends to be an association for chromosome 16 of these distally positioned crossovers that hold homologous chromosomes together, whereas for something like 18 and also 21, there's a very high preponderance of what we call crossover left or achiasmatic chromosomes, where the two chromosomes from the mother failed to cross over and form a connection. So why are these connections so important? Well, we think they're important because the crossovers give rise to these chiasma here that you can see. This is an EM of a grasshopper spermatocyte. This is just a schematic we've drawn. And these bivalent structures here held together until the cell cycle cell cycle progresses to anaphase when these cohesin molecules here basically cleaved and the two homologous chromosomes separate. So without these physical linkages, the cell has no idea what it should be dividing. So in essence, if there are no crossovers and no chiasma, the cell will find itself in a situation 50% of the time where it divides one to the same side, as shown here by these fish. This is budding yeast. And in 50% of the cases, it'll take both chromosomes to one side, as shown here in fish and yeast. In budding yeast. So having crossoverless chromosome pairs is really bad. And it's particularly bad in human oocytes, because in human oocytes, oogenesis takes decades. So in sperm, it takes about 40 days. In budding yeast, we can do it in 8 hours. In human, it takes a lot longer. So the chromosomes start pairing. They form these bivalent structures when the woman is a fetus undergoing fetal development, taking place there in the ovaries. And these structures are formed by about 18 to 22 weeks. Then the oocyte enters a prolonged state of arrest here. It's called G2M, but it's called dictuate in this case here. And then when the woman starts maturing, 10 to 50 years later, once a month, a single oocyte will then undergo meiosis 1, where the homologous chromosomes is separate from each other, one into the oocyte proper, one into the first polar body. And then upon fertilization, the two chromatids will then separate. So this takes decades in human. And at this arrest point here, it's absolutely essential that the two homologous chromosomes are well connected. Okay, so even if they form a connection, however, over time, it is thought that this structure here might be depleted, giving rise to these kind of aberrant structures here, via cohesin loss, for example. So you could imagine that, or we could envision that cohesins along this arm here is lost, giving rise to what we call univalence. Loss of cohesin between sister kinetochores would lead to their precocious separation, a meiosis 1. And you could also envision that entire sister chromatids might come apart. Okay, so what we're interested in the lab is to understand how achiasmat chromosome pairs without physical connections arise. So I'm going to focus on how fluctuation and variation in recombination and the generation of crossoverless chromosomes occur in the human oocytes. So the first thing that this kind of model kind of suggests is that women, some women might have fluctuation or variation in their recombination rates, and that this may lead to crossoverless chromosomes in some of their oocytes. So the first thing that we looked at was basically whether there is variation in this, and that's indeed the case. So what we do is we get oocytes from human fetuses and or ovaries, and then we spread out the individual oocytes, and we stain them with a marker that we can follow the actual chromosome, shown here in red. Blue shows the centromere of each chromosome, and then we look for these dots here, these foci of MLH1, which is a molecular marker for where the crossovers are. And we then have algorithms that allow us to determine how many foci there is. And here are 6 different individuals that we've studied, and you can see here that the number of MLH1 foci or crossovers are very, very different between the individual, but there's also substantial variation with what we see within individuals as well. So it certainly appears that we have tremendous fluctuation and variation in recombination between women and also within women. So the question is, are there crossoverless chromosome pairs in human oocytes? So again, what we do, we stain them as before, and now what we do is we look for chromosomes that fail to show an MLH1 focus. And you can see this is the case here. You can see this is the case here, there's no green dot. And then what we could do is we go in and we follow that with fish, and we ask what chromosome is it. So for the ones we have fished against, you can see here that chromosome 21 and 22, the two smallest chromosomes, apart from Y, are the ones that preferentially seem not to get a crossover, or they become crossoverless. However, overall, what we see is that 20% of human oocytes contain at least one crossover-less chromosome pair. So this is a really vulnerable oocyte. And secondly, the crossover-less chromosome pairs, they occur predominantly on these smaller chromosomes here. Now, when we can compare that to what we can do with sperm, in sperm we see fewer than 1 in 1,000 sperm that have a crossover-less chromosome pair. So, so what we thought next was basically the reason that we're seeing these crossover-less chromosome pairs predominantly in human oocytes as opposed to human sperm might be because perhaps women have lower recombination rates overall than men, than in male meiosis. So, for example, there are fewer crossovers to go around. This wasn't the case, however. We were quite surprised by these findings. So if you look at the male distribution, they're quite tightly distributed in the sperm, and in the female they're much more spread out, but there's certainly a lot more crossovers. So that doesn't seem to be able to explain why in women of female meiosis there's so many more crossover-less chromosome pairs. So what we then considered was the idea that do lower recombination rates in women oocytes, so the ones here, do they actually lead to crossover-less chromosome pairs? Does the formation of crossover-less chromosome pairs have anything to do with low recombination rates and therefore lower probability of getting one? So we're looking at this distribution here in the women, and what we find is that overall the risk is about 20% that any given oocyte has a crossover-less chromosome pair. But when we look in this population here that overlaps really nicely with sperm, you can see that we see an increased threefold risk. So we see a 62% risk of the oocytes that overlaps here with the sperm in having a crossover-less chromosome pair. So the implication for this is that in women, in female meiosis, the distribution of crossovers amongst chromosome pairs is just much less well regulated than in male meiosis, okay? So it seems that in female meiosis, one strategy is to just simply flood the cell with crossovers and hope that everyone gets one. Okay, so what may determine the recombination rates that we see, and why is this so variable? So what we did was we looked at 40 different fetuses, and what we found was really quite intriguing. We found that when we looked at the mother who had lost the fetus, the age of the actual mother carrying it, that there was a significant contribution of her age to what was happening in her daughter's oocytes. So this is kind of intriguing because it suggests that we're having an intergenerational or transgenerational effect of recombination rates. So when we look at the mother's age carrying her daughter, and you look at the recombination rates in the daughter, the age of the mother actually matters for what happens in the daughter. Okay, so when her daughter is born, her recombination patterns have been set because recombination took place at this stage here. So when the daughter then chooses to start her family, she may suffer infertility, and the grandchild's karyotype might be affected because her grandmother was older when she carried her, the mother, basically. So I think the conclusion of what we're seeing is that we have some transgenerational effects in maternal age and aneuploidy. The contribution to the variation is about 5%. And we also see intergenerational effects in maternal age on the daughter's reproductive potential. This is called the grandmother effect. Okay, so I think what I've shown you for this section here is that indeed, there are low levels of crossovers in human oocytes. Recombination events, lead to crossover-less chromosome pairs, and that the mother's age influences what happens in her daughter's oocyte, eventually affecting the grandchild. We also in the lab looking at whether vulnerable crossover configuration with age may lead to these achiasmatic chromosome pairs, as well as whether this kind of model here actually applies to human, as it does in a variety of model organisms that we've looked at in the lab. Okay, so With a little bit of time left, what I'd like to do is just to showcase a bit more of science that we're doing in MRC Genome Center, and also within the wider School of Life Sciences at Sussex. So I'm just one of many. There are about 20 groups now. You've heard from Keith and Alan, who are some of the most senior people. Alan, a former chair, Keith now deputy director. We have a number of very young scientists, and we're looking at this molecule here, DNA, and various aspects of DNA repair in developmental disease disorders, cancer, etc. The MRC funds part of partners with the University of Sussex in having the MRC Genome Damage and Stability Center. The MRC's mission is to encourage and support high-quality research with the ultimate aim of improving human health. Part of that remit is to produce skilled researchers as well as technology development, and also engaging with the public about what medical research is actually important. With respect to technology in particular, I want to spend a few minutes showcasing some of the technology we use for our science in the Genome Center and the School of Life Sciences more generally. This is a quote by Sidney Brenner, that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And I'm going to show you that that's probably the case. Okay. So since the MRC started a century ago, we've had tremendous development in my favorite technology area, which is imaging. And when you look at what they had in 1913 to help them do biomedical research, they had X-rays, which had been developed in 1895 for medical purposes by Röntgen, and this is a the first Röntgen photograph of his wife Anna's hand, and they had standard light microscopes to look at specimen that they could see, not very well. In the 30s, we saw the development of electron microscopes, so we were now in the picometer resolution range with transmission electron microscopes. SEMS as well was developed by Arden in 1938. And in the 30s and 40s, biological or biomedical scientists started using fluorochromes that have been first described by Stokes in the 1850s to start conjugating, using them in the life sciences and conjugating them to antibodies, so you could now look at fluorescence of specific proteins of interest that were being detected by your specific antibody. The real development with respect to following kinetics and dynamics of cellular processes occurred with the development of fluorescent proteins by Shimomura in Japan in the 60s and 70s from the jellyfish A. victoria, where he purified aquarin and the famous green fluorescent protein, characterized them. And it was in 1992 that an American scientist realized that, or first cloned it, gave it to a number of different labs, and the Chalfie lab was the first to express it in E. coli and C. elegans. Now that, together with development in CCD cameras, computers, LED, and lasers, has really facilitated the technology development that allows us to follow cellular processes in real time and understand biologically important processes. In the late 1950s, the development of confocal microscopy started as well. As opposed to seeing all the light coming through the microscope, this allowed scientists to focus on just the plane that you were interested in. So this left is what you would see normally of a pollen grain, and this is a confocal image. You can really see the crispness and the sharpness in the axial dimension in the set plane. And then finally, in the noughties, we've started seeing the development of what we call super resolution microscopy, where we're beating the resolution of light. And I'm going to show you an example of what we're doing with that within the School of Life Sciences. Finally, the MRC in particular has been instrumental in implementing and developing both PET and MRI full body scans that's used in cancer research and neurosciences. The MRI basically funded Sir Peter Mansfield, who was instrumental in using and developing mathematical equations that lowered the imaging time in MRI machines from literally hours down to seconds, so that MRI scans became feasible. With respect to PETs, which relies on radioactive traces, The MRC were the first to realize that this was really important, and they installed the first cyclotron at a hospital site in 1955 at the Hammersmith Hospital in London, so that the physicists and the actual clinicians were in the same, not in the same room, but certainly next to each other. Okay, so within the Genome Damage and Stability Center and School of Life Sciences, the way that imaging is changing the way we work is, I think, very well demonstrated by Velebo Savage, who is using Widefield imaging for high-content screening. And what Velebo does is use siRNA libraries. He puts in his cell line of choice, or your cell line of choice, and then he images whatever processes automatically you're interested in, and you get your output in this kind of format here, and you can then go and analyze your data and look at which hits might be interesting. Now, Helfried Hochegger is interested in mitosis, and he works with John Atack, who's talking tomorrow, as well as Simon Ward in a drug discovery department. And he's interested in finding targets that are important for mitosis, the cellular process that you see here. In red, you see the tubulin. In blue, you see the chromosomes. And you can see the cell here undergoing anaphase, followed by cytokinesis eventually. This then allows him to start screening for drugs that inhibits mitosis and that may eventually lead to useful anti-cancer treatment drugs. Another way that imaging and the technology development has facilitated research is this XP variant polymerase eta here from Alan Lehman's group. And you can see after UV radiation, pol eta here accumulates into these foci that coincide with where replication is taking place. Okay, so Mark O'Driscoll is another young scientist at the Genome Center, and he works with clinicians on rare human disorders and genome instability. And you can see here with his clinicians, they use MRI scans to diagnose this microcephaly disorder. And you can see that the brains are smaller, it's detached from the skull, there are black spots here, and it's way too, the contours of the brain are way too smooth. What Mark then does is he gets the cells from these patients, and together with exome sequencing, he tries to then look for cellular defects that may be associated and therefore causative of the defects. And in this case, the exome sequencing identified STAMP-BP, which is important for removing these ubiquitin chains as receptors on the surface are internalized. And he found that if you inhibit, if you knock down STAMP-BP, you get this accumulation of ubiquitin chains in the cells, and you can see that the patients are showing these kind of accumulations as well. Okay. So this is Tony Carr, who is our director, who is unfortunately not here. He works with Mark O'Driscoll in the chemistry department, his postdoc as well, and we have an informatician, Alex, and they've set up and built from scratch a super resolution microscope that allows us to look at single molecules in real time. So very basic kind of, I guess, introduction into this is that when light is diffracted onto the camera, there's a kind of a blurring effect, as you can see here. This follows the Bessel function, and it means that the response of an imaging system is only as good as the point spread function. So that means that the resolution becomes the minimum resolvable distance between two objects in the image. So you can see here to the left that these are very nicely, the peaks are very nicely separated. You will see these as two objects. But as they become closer together, you can see they start merging, and you can't tell whether that was one or two molecules. And for light microscopes in the XY axis in the plane, this is about 220 nanometers. So how do you overcome this? And this started in the 2000s. There are a number of ways, but the way that Tony and Mark have set this up is to use photoactivatable localization microscopy, basically. And what that does is that you tag your protein of interest to a fluorophore. It's in a non-fluorescent state initially. Then you activate it with a laser, it fluoresces, and then you image it, which causes it to bleach, basically. For storm, it goes into a dark state. Now, if the activation is short enough, you'll only activate a fraction of your molecules, as shown here. Okay, you then shine the laser again, and you get some different ones stochastically emitting light. And then at the very end, you take your image reconstruction and you build up an ensemble image of where your individual molecules were in the cell. Okay, so this is what it looks like in real life. This is PCNA, one of Alan Lehmann's favorite proteins. And the raw data here, and this is the fitted, these are the fitted data. These are single molecules emitting light that you're seeing, basically, of PCNA. So, how impressive is it? Well, these are the Pombe cells, efficient yeast cells that Tony uses as a model organism. They're ideal for palm because they're very flat. Now, under standard microscopy that's diffraction limited, this is what you would see. Okay, on a microscope, if you just fix your cells and you look down, that's what the image you would get. But with the super resolution palm, you see these structures appearing, these dots, and I've just blown them up here for you, enlarged, so you can now see individual molecules, you can see clusters as well. So the resolution we are at the moment is down to about 50 to 100 nanometers as opposed to 220 nanometers with conventional microscopy. And we're hoping to get down to about 25 nanometers. Okay, so there are a couple of interactions within the school with the Genome Center that I just wanted to emphasize. The MRC doesn't just fund the MRC Genome Center in collaboration and partnership with the University of Sussex and School of Life Sciences. The MRC also funds individuals and projects throughout the school, basically. And particularly within biochemistry, there's some BSMS funding, and in particular in neurosciences. So I just have a couple of slides I wanted to show you for people who can't be here today. This is Majid Hafsetsperes, and Majid is interested in motor-neuron disease that Keith briefly touched on. Now, motor-neuron disease, as Keith explained, is not only caused by genetic defect in DNA repair genes, but also in dynein or motor protein defects, genetic defects therein. And what Majid does is he has built up some mouse models, and then he can track the dynein activity by following these endosomes here in real time, that you're following a single endosome moving. And you can look at basically whether or not that trafficking is affected or not. Finally, Leon Agnado is the new, new head of neurosciences at the school. And what Leon does is he's interested in how synapses contribute and how they process visual stimuli at the retina. And he uses zebrafish as his model organism. So this is a zebrafish here. This is just a fixed image of the inner cell layer, inner layer of the retina where the synaptic activity takes place. He can take the fish, he can put it under confocal microscope, and